This is a method to apply electromechanical stimulation on cells. It has multiple applications to study its effects on a cell population, pre-training for in vivo delivery, and cell maturation.
The advantage is that electrical and mechanical stimuli can be applied with the same device, individually or simultaneously, while keeping the sterile barrier intact.
This technique is an indirect approach toward therapy. If cell therapy is considered, electromechanically stimulated cells may be an interesting cell population to treat injured myocardium.
This method genuinely belongs to the cardiovascular field, but it could also be applied to the nervous system.
This is an easy technique that requires patience. The most difficult part would be working with the small pieces and keep the sterility throughout. There are little details of manipulation which are difficult to explain, so the visual demonstration is really helpful.
Begin by transferring 12 cleaned PDMS constructs to sterile 10 cm plates. To ensure complete sterilization, expose the plates to UV light for 5 minutes. Then transfer each construct to a separate 35 mm cell culture plate or 6 well plates for immediate cell seeding. To start cell seeding, first wash a Confluent T75 flask of cardiac AT-DPCs with 5 ml of 1x PBS. To detach the cells, add 1 ml of 0.05% trypsin EDTA and incubate at 37 degrees Celsius for 5 minutes. Then add 5 ml of complete medium to inactivate the trypsin EDTA. Collect all detached cells in a 15 ml tube. Wash the cell flask twice with 5 ml of